All right. Hello. Thank you all. Appreciate the opportunity to speak here. The last Nextflow event I went to was in 2019. And so I've been doing Nextflow and NF Core since before it was cool. I've been doing bioinformatics for almost 20 years. So it's a great opportunity to be here and share what we're doing here at Seanome. So my name is Olga Botvinnik. I am the CEO and founder of Seanome, which is a nonprofit research institute for marine genomics. What we do is we build open source software to figure out how the genomes of weird creatures in the ocean can help us live longer and healthier lives. Yes? Having trouble advancing. Oh, because these are my slides, duh. Okay, great. That's why it's not working. Awesome. So first I'll introduce what Seanome is and why we at Seanome are annotating proteins of unknown function, and talk a bit about the protein annotator pipeline and some hot off the press updates from the hackathon this week. So what is Seanome? It's the coolest nonprofit research. Oh wow, great. It's my talk. Nice to know. All right, Seanome is a nonprofit research institute for marine genomics. It's the coolest nonprofit you've never heard of. We were incorporated last year in October and have officially received our 501c3 designation. And so far, our mission is to unlock breakthroughs from ocean biodiversity by building open genomics tools. My vision is to open source the code of life so we can start living in harmony with nature. So what I mean by that is a really broad vision of breakthroughs, not only on the disease side and therapeutics, but really thinking broadly, what if we were able to build our cars out of seashell-inspired materials? What if we were able to live in a built environment that is biologically driven and filters our air and is more integrated with our nature's environment? And I think by learning how nature already creates structures and creates proteins, we can create that environment, and that's where I want to build towards. What we do, as I mentioned, we build open genomics tools to annotate proteins of unknown function. So we're a combination of three main focuses. So one is protein functional annotation. And as I mentioned, we are developing methods and writing papers for protein functional annotation. We are developing open source software. For me, the main output is the software and is making tools that are useful for others. And we're focusing on the ocean and marine animals because I believe the ocean is the most undiscovered part of the world that we possibly have. Almost 80% of life on Earth lives in the ocean. So the amount of biodiversity we can see there is just huge. If you think of animals that can't live on land, like jellyfish or octopi or whales, and what we can learn from their biology to understand better ourselves. I think we can learn really deep biological truths. And for example, a bit more on the ocean is that in the ocean animals, we have around 43 billion proteins, and only about 8,000 of them are known. And yet already our 13 FDA-approved drugs worth $8.5 billion have already been found from organisms in the ocean, some even from the same organism, the same sea sponge, Tryptothera cryptica. It has had both an HIV drug and an anti-cancer drug. So if that's just one organism, think about what we could discover from the rest. So I want to emphasize that because biodiversity is really our main frontier, the biggest frontier I see in the world. Ozempic, for example, was originally derived from lizard venom and is now a huge blockbuster drug. So I think by focusing on biodiversity and the biodiversity of the ocean specifically, which has the greatest biodiversity, we can make a really big difference. And in particular, so why we're nonprofit is we are developing open source tools because I want to make science better for everyone. I can't possibly discover all the cool new therapeutics or cool new patents to make the world a better place, but I can enable the most scientists to do so. And so far, we've raised our first $25K. I should mention we also have about 10 volunteers who've already contributed over 350 hours of their time of donations in kind, and I've been really grateful to work with them. We build on the strengths of other organizations. So like academia, we also do basic research and do publications. And in academia, because the focus on marines and open source development can be a bit mixed, that's where we differ. On government philanthropy, such as NOAA and Schmidt, other organizations can be very, very focused on marine genomics and the ocean biodiversity. And then our focus on open access, which is beautiful, but the tool development and engineering culture is a little secondary and can be a bit more slow-moving. So in AI biotech, such as DeepMind and Recursion, as Evan mentioned earlier, these are organizations that are using protein sequence to see how they could be used better for health. But the models are often proprietary for profit, but we do really like the tool development and engineering culture at these organizations. Now, why annotate proteins of unknown function? I want all of you to pull out your phones. We're going to do a quick poll here about how many of the 20,000 human protein coding genes are of unknown function. We're going to do a quick poll here. If you're at the hackathon, you actually have the answers, so that's why you should go to the hackathon next time. Maybe you learn extra things. Do 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 do. Okay, great. 15 participants, awesome. 10,000, so half the human genome, unknown function, 5,000, quarter of the human genome, 2,000, 10%, 2,500, cool. All right, so we have some people who really think we don't know anything about the human genome, which is great. I love the skepticism. Oh, wow, 54, amazing. Thank you so much. Great, so we do know a bit more about the human genome. About 1 in 10, or 2,000 genes in the human genome have unknown function. And I wanted to highlight this because for people who are working in human systems, especially for clinical work, it's like, well, why do we need to annotate proteins of unknown function? We already know what P53 does. It's like, yeah, because that's what the literature is biased towards, is the proteins that have already been studied, especially in humans. So I just wanted to highlight that when we're thinking about proteins of unknown function, even in human, we still don't understand what all of our genes do. And when you come to isoforms, like, don't even get me started. That we're just starting to understand. So the problem is the vast majority of proteins on Earth have unknown function. So we have 14 quadrillion predicted proteins on Earth. So quadrillion is the one after trillion, which is the one after billion. And that is a really, really huge number. So of the proteins that we do know what they do, we have about 570,000 in SwissProt, UniProt, that we understand that have some known and manually curated function. So we talk about what we know and what we've seen, it's far less than 1%. Like, that's not even an adequate statement, as we know less than 1%. We know like millions of times less than what is truly out there. And to orient you a bit here, I used my cursor. Okay, great. So this is a dot plot on the left, so each dot is one species, and the size of it is the number of proteins of that species in this database. So in UniProt, SwissProt, we have some species like human, mouse, Arabidopsis that are biased, that make up the majority or have a big blob in the database. So when we train AI models on databases that are biased, that means our results are also biased towards what we've seen before. So if we really had a database of all predicted proteins on Earth, then every species would be equally represented based on its proteome size, not based on whether we've seen it before, like it or not. And I just want to emphasize this known protein universe is limited and biased by many 11 orders of magnitude. So on the first column here, we're looking at proteins with a high quality structure. It's about 67,000 proteins are in PDB. This is what AlphaFold was trained on. Of that, we have 570,000 that are understood. This is from the previous slide. From UniProt, SwissProt, then cataloged, we have proteins in UniProt Tremble. So this is proteins that are in a high-quality database from UniProt. And this is if we really look at all the proteins that we know that we trust are from real data, that's about 250 million. But the difference in order of magnitude from what we understand and what's really out there is 11 orders of magnitude. Again, 1% is like not even close to explaining how little we know about biology. So we need tools to bridge this huge 11 order of magnitude protein knowledge gap. But what about AI? It's supposed to fix everything. So I want to pause here because as we're building here, unlike written language where we can observe everything that's been written down, we can read the Bible, we can read Wikipedia, we can read Reddit, we can read every text that's ever been out there. But of all organisms, even that exist currently on this planet, we have not observed their biological sequence data. So it's as if we've trained ChatGPT on Russian math textbooks. And now we're like, hey, write my emails, write a legal document, write a contract. Like, it just doesn't have the right training data. Write code. It doesn't have the right training data. It's like you're learning on an alien planet. You've learned your languages on an alien planet, and you are now on this alien planet, and you're like, oh, I'm hungry. Let's type restaurant. But you just don't even know the word for restaurant on this alien planet. So marine genomes, and really the majority of genomes on this planet are alien to what we know so far. So just to drive this home, but why I'm an AI skeptic. AI is really only as good as its training data. As I mentioned, AlphaFold 2 was trained on the PDB structure, so really 67,000 proteins that we know so far. It is a lot, like really, really beautiful effort from crystallographers in the world who have contributed and made this data available, but still not everything that we have seen. Another tool called ESMFold, if you're following the AI literature, they did a DeepSeek-like approach where they trained their model. They were able to get away with lower compute and resource capacity by training on the hallucinations, aka predictions, from AlphaFold to do their protein structure prediction. So we need methods that are sensitive to detect novel function when the sequences are quite different. So our solution with Kmerseek at Seanome is to develop a tool called Kmerseek, and I'm happy to tell you more, but effectively what we do is use a binary amino acid alphabet to increase our effective database size, so we have many more matches than other tools, and including AI tools. And because we're using computational tricks like Kmer hashing, our resource footprint is 1,000 times lower than AI. So happy to tell you more. So back to Nextflow and NF-Core. We at NF-Core are building the best protein annotator in the world. As I said in the hackathon, we're going for world domination here. And we're bundling Kmerseek with other tools. I think it's very important to show that our tool will shine in certain cases, and every tool has its own strengths and weaknesses. So what we're doing is we want a win-win-win for the community so that a community member can come in and say, hey, I have this cool new weird creature that I've sequenced. What do these proteins do? What are their functions? Where do they localize in the cell? What do they bind? And I'm showing you our initial metro map. So we started at a hackathon drawing the metro map first. And a big challenge with these tools is that each tool requires its own individual unique database. So if you've done RNA-seq alignment, you know that that is a big 60+ gigabyte database that you need index to. Imagine you need that before every the 20 tool that you need for protein annotation. That's what we have here. And a secondary challenge is that some of these tools are wrapping big bash scripts that really could be their own Nextflow pipelines and doing some duplicated work. So a bit of 2 hackathons. So we started in San Francisco. Thank you to AWS and Heritage Bank for sponsoring. We started our work on protein annotator, adding 4 tools to the pipeline. This week, we've continued our NF-Core protein annotator work, adding 4 more tools, and migrating to NF-Test, which was super exciting, and expanded Unifier to a sub workflow. So thank you especially to Addie, Chase, Jessica, and Saba for all your hard work. So here's the progress so far. We started with this metro map. We now have a lot more tools. All of them are still in progress. A big piece of what we did was migrate to NF-Test. And now NF-Test is really the red pill that you're taking to go into the truth of the data that you're finding, since an Nextflow run profile test is it's a great thing to have, but still equivalent to a smoke test. Does this pipeline run or fail? But the NF test, you're actually testing for the truth, quality, to what you found originally. And now passing all the tests is like, yay, I'm going to pass all the tests. It's going to be amazing. This is going to be the best pipeline ever. And then you're like, oh no, all the NF tests? Really, all of them? How am I going to get there? So just wanted to share some of the trials and tribulations in building some of the pipelines and how we really love the NF test with a new framework to build there to get at the truth of the reality of what we're testing. But we're all learning in this together. And with that, I would love to pause and for any questions, and thank you so much for your attention.
All right, thank you very much. That was a really cool talk. I'm pulling up my Slack questions. Does anybody in the room, we're gonna start with the questions in the room on this one. Anybody in the room have any questions? No? If not, I had some questions about how do you, are you working with any organizations to do the part that goes in and goes and finds organisms and sequences them?
Yeah, I forgot to mention, so we're starting with a collaboration with my colleague who has been working to cure his father's neuropathy for over 15 years. And he has convinced the Canadian and Norwegian governments to go diving for Arctic clams because Arctic clams have a unique ability to tolerate this neurotoxin that in humans causes neuropathy. So we want to figure out what's different about the genome from the Arctic clam. But in general, Seanome is purely computational, and we are working with partners for both sample collection and validation, because ultimately, computational work is a prediction. We have to go to the lab and see, does this actually validate in the lab? Do we see this in a cell-free assay, in a cell assay, in an animal, and so forth? But yeah, so we have to work with biologists. We can't work in a vacuum on computational work.
Yeah, that's fantastic. I mean, that's the collaborative approach there is really fantastic. Harshal asks, if we don't have good training data due to very few proteins being functionally annotated, what proportion of proteins on average from new species can we actually functionally annotate? And how best would you identify novel functionality?
Sure, so I can give an anecdote from our recent Arctic clam data. So we had 30,000 genes from the Arctic clam data, and 2/3 of them were able to be annotated by BLASTP, simple annotation tools. So another 10,000 of them were of unknown function. And with Kmerseek, we were able to annotate 45% with an additional, some initial parameters, and I expect us to reach 90% and more soon.
That's amazing. Ken asked, what is your favorite weird sea animal? Is it the Arctic clam?
Oh, Batrillus schlosseri, a colonial tunicate, is near and dear to my heart. It was what got me into the ocean annotation game, and it was the first prototype that I wrote for Kmerseek, where this organism has a gene that's really similar to our HLA transplant rejection genes, where it has to have the exact same allele for if you or I needed a bone marrow transplant to have a match. And yet this protein has no similarity to any human proteins. And every tool just completely failed on finding anything on it. InterProScan was like, ooh, there's distorted protein regions. And I was like, great, that doesn't really help you know what this does. And so with Kmerseek, we were able to find 6 domains that then allowed the experimentalists to have actionable results about new data that they could dig into. And some of them were also experimentally validated.
Very cool. Any questions in the room? If not, thank you very much. I'm sure there'll be follow-up on Slack.